So today we're going to focus on elements of life and properties of biological macromolecules. Biological macromolecules are organic compounds from your ninth grade year. So this hopefully will be mostly review. Again, remember, pause, rewind, whatever you need to do to get the information down. Also remember, what is said is just as important as what is written down. Okay? I will say more than what is written down, because there's just not room. It gets too messy, it gets too hard to follow. So make sure that you're paying attention to what is said and not just copying what's on there and stopping. Also remember that diagrams are your friend. You may sometimes want to pause and either copy some of the diagrams or draw your own diagrams, but diagrams and pictures will help you a lot. Okay, so let's talk about some of the elements that are important for life. Okay, I know you, again, most of you are currently in chemistry. I'm not expecting you to memorize the periodic table. We're not even really going to look at it again. Okay, I just want to kind of remind you that these are elements. Okay, we've got carbon, nitrogen, oxygen, okay, hydrogen, phosphorus. These are all important elements down there. Okay, these are all important elements that are essential for life. Okay, to build molecules that can sustain life, these are the basic elements that you're going to be using them. They're not the only ones, but they're the most common that will be contained in those molecules that can contain life. And in order to maintain organisms, you have to exchange these with the environment. Okay? There has to be new carbon has to be brought into the organism, new hydrogen, new nitrogen. Okay? And so these elements are constantly cycled between the organism and the environment around them. So we're not going to memorize biogeochemical cycles. Remember from your freshman year, we talked about carbon cycle and nitrogen cycle, and we interpreted some pictures of those. But that's what we're talking about. Those are what we're talking about here. And we're talking about biogeochemical cycles. Carbon cycle, nitrogen cycle, phosphorus. Okay? That's what allows for the exchange between biotic and abiotic reservoirs. Remember, biotic are going to be the living organisms, where the abiotic would be the non-living reservoirs, the rock, the fossil fuels, okay, the atmosphere. And so there's constant exchange between these abiotic and biotic reservoirs. Nutrients in our living organisms, okay, so you can see right here, nutrients in the living organisms are, and in detritus is dead organic material, is available to other organisms, okay, when they feed. So when a herbivore comes and eats a plant, they're going to get some of the nutrients there from that organism. When a detritivore consumes the non-living organic matter, so the dead, broken down, non-living organic matter. When a carnivore comes and eats the herbivore, okay, there's nutrient exchange that's happening there. Okay, some of this will be released. Some of these nutrients will be released during erosion, burning of fossil fuels, weathering. Some of it is basically put into storage, put into the inorganic reservoirs, okay, during fossilization. Okay, when you are sedimentation, when we're forming the rock. So any of that material cannot be assimilated easily. You guys will see that term a lot. Okay, assimilated or assimilation. Basically, it's brought into the organism. It's made a part of the organism. You can see right here, there's your assimilation. Okay, and so if the organic materials have been fossilized, if they've gone through sedimentation, you're going to need something to release it from there. Those cannot be easily assimilated into the living organism. Okay, we need to release those into the atmosphere, into the soil, into the water. Decomposition will help release some of these materials to allow for that assimilation, right? Our plants can assimilate things from the soil, okay, and then that can pass through the food chain. But there's this constant cycling happening between living and non-living reservoirs for these different nutrients. Okay, let's start with carbon because all of our organic compounds have carbon in them. Okay, carbon is you're going to have to get it from the environment. We're going to need it for atoms and molecules, and we're going to need to be able to get it from the environment. We use it to build carbohydrates, lipids, remember fats, proteins, nucleic acids. It's going to be used for storage molecules. It's used for cell formation. It has a wide variety of uses. And the reason why is it has the ability to bond to 4 atoms. If you look at the valence shell of carbon here, right, that outer layer here, it has a valence of 4. So it currently has 4 electrons and it needs 4 more to be happy. So carbon has the ability to bond with 4 atoms. They could be 4 different atoms. They could be 4 of the same. They could be a mix of a couple. Okay? And so its ability to bond with 4 different atoms allows it to have a lot of diversity in the different kinds of molecules it can build. It can build molecules that are shaped like rings. It can build molecules that are in straight lines. It can build molecules that branch off these straight lines. It can form double bonds easily. Okay? Double bonds allow for bending within molecules. So there's a lot of the structure of carbon. So again, coming back to, like we did with water, the structure of these molecules and atoms are what are determining its function. And so the way carbon is built, with the fact that it has a valence of 4 and the diversity of molecules it can build, allows it to be kind of our major stepping stone into other molecules. So nitrogen, okay, needs to be again pulled from the environment. It builds proteins and nucleic acids. Remember, our nitrogen bases are amino acids for proteins contained nitrogens. Nitrogen has a valence of 3. That's a typo. Nitrogen has a valence of 3. It currently has 5 valence electrons. It needs 3 more. So it has a valence of 3. Okay. Remember from 9th grade again, nitrogen is something that we as animals need to consume in our diet. Plants assimilate it from the soil, right? Remember, plants will take it in from the ground, from the soil. Remember, there's lots of nitrogen in the atmosphere, but that's nitrogen that we can't use. So we need the bacteria to fix it in the soil, and then the plants will assimilate and take it up, and then the herbivores eat the plants, and the carnivores eat the herbivores. So nitrogen has to be consumed in the diet, or in the case of the plants, assimilated from the ground. Phosphorus is used to build nucleic acids. Remember our sugar phosphate backbone? So used to build nucleic acids and some lipids, in particular phospholipids. Remember, phospholipids are found in the cell membrane. Okay, and they contribute, the structure of those phospholipids is what determines the plasma membrane's job. Its ability to allow some things in and not other things is due to its structure, due to the fact that it has this phosphate on those lipids. Okay, and so it's built a little bit differently. So phosphorus is another molecule that is important for these, another atom that's important to build these organic compounds. So to build these organic compounds, we're going to have to do some chemical reactions. And we're going to want to both build and break them down. Okay, so remember our vocabulary from our freshman year, right? We have polymers, those long chains of molecules, lots of monomers put together. Okay, and remember the monomers, they're the subunit, they're the building block. Okay, for example, this is a very, very small polymer here. It's a disaccharide. Okay, most polymers can be 10 monomers long. They could have thousands of monomers. Okay, so we've got a very small example here. Okay, and if we want to break this polymer into the monomers, okay, which we do quite a bit. Okay, the most common way we're going to break these polymers down into monomers is we're going to add water. And when we add water, that allows, you see there's one hydroxyl group went to one of the polymers, and then the hydrogen atom went to the other polymer. Okay, and so, went to the other monomer. So adding the water splits the bond here, holding the polymer together. So if this was a really long polymer, let's say we had more, that's a terrible ring, but you get the idea. Say it was a really long chain, water would have to be added to every bond between the monomers here to be able to break them. We'd add water each time. Okay, and that would then add a hydroxyl group to one and a hydrogen group to the other monomer, making these stable. This is glucose and fructose, so making completely stable monomers there. That reaction is called a hydrolysis reaction. Okay, hydro is water. Remember, lysis is to break or to burst. So I'm using water to break these molecules apart. This is sped up by enzymes. Remember, enzymes will speed up chemical reactions. So this process is sped up by enzymes when we add the water and we split the polymer into the smaller monomers. Okay, this is very common. Let's look at an example of this, digestion. Right? So think about us as humans. We do digestion all the time. It wouldn't be a surprise to me if you were eating a snack right now. Right? And so as you take in that food that you take in, it's in a polymer form, right? It's a piece of meat. It's a chip. It's a carrot, whatever it is. It's in polymer form, right? Lots of little monomers put together. But let's say you eat a piece of chicken, okay? So chicken is protein, right? We all know that. Chicken is protein. I'm eating the muscle of a chicken. That's where I get the protein from. So I'm eating the muscle of this chicken. Okay, well, why am I eating it? My body says I need protein. But do I need chicken muscle protein? No, I don't need chicken muscle protein. I have human muscle protein. But what I need are the pieces to make human muscle protein. And the chicken protein contains those pieces. So when I eat the chicken and the process of digestion is happening, these hydrolysis reactions are happening. I'm breaking the proteins into smaller amino acids. I'm breaking them into the monomers. And then I can reassemble those amino acids however I want to make the protein that I need. Okay? So these hydrolysis reactions are really important to us. On the opposite end of that, sometimes we want to take those monomers and build them into bigger polymers. Okay? For example, when you have digested that chicken protein and now you have all the little chicken protein pieces, but you need to rearrange those pieces into human protein order, I'm going to build a polymer. And when I do that, I'm actually going to take away a water. Okay, so if we look at the example that we have right here, these are amino acids. This is a protein example. Okay, so I have one amino acid on this side and another amino acid on the other side. The way you will be able to recognize amino acids, and this is something you should probably just file away, is they have this repeating NCC pattern. NCC. Okay, you'll notice actually if you look closely at these look the same. Okay, they both have that carboxyl group at the end, the carbon with the two oxygens on it. Okay, they both have a side chain, which that is the only thing that's different from amino acid to amino acid. That is what makes each amino acid different from the other, right? They have the NH3, they have the amino group, they have a hydrogen. Okay, it's the R group that makes them different from one another. So if I want to combine these two amino acids to start forming my protein, okay, what I will do is I can remove a water. I can remove, okay, if I remove the oxygen from here, from this carboxyl group, okay, and I remove two, see how this is an NH3? I remove two of those hydrogens. You can see there's your hydrogen still there. Okay, I remove two of those hydrogens. That gives me a water. And now I can form a bond between that carbon and that nitrogen, and I get my peptide bond. So every time when I'm making new proteins, I'm doing this over and over and over again. I'm going to remove this one. I'd have another amino acid over here, right, that I would remove the hydrogen. I'd remove 2 of the hydrogens from to make a peptide bond between those. And I would do that all the way down the chain until I was done making my protein. Okay, so these are going to require energy. They're going to require enzymes. Think about that logically, right? It's going to take a little bit more energy to build something usually than to break it down. But both of these are important, and our bodies, all organisms, are doing this all the time. Even little one-celled bacteria are breaking molecules down and rebuilding them into the ones that they need. Okay, and so these are constantly happening. So let's look a little bit closer at these organic compounds. We're actually going to start with nucleic acids. Remember, nucleic acids are DNA and RNA. Those are our 2 examples of nucleic acids. So from freshman year again, let's run through this really quick, right? Our monomer was the nucleotide, right? A nucleotide contains these 3 things, right? A nucleotide contains a phosphate, a pentose sugar, right? What's a pentagon? 5, right? So it's a 5 carbon sugar, deoxyribose or ribose. So deoxyribose I'm going to find in DNA, right? And ribose I'm going to find in RNA. So we're going to look, just a reminder of some of our differences here, right? And then we have the nitrogenous base, again, all part of our nucleotide. There are 5 nitrogenous bases. There is cytosine, guanine, adenine, and then DNA has thymine, where RNA has uracil. Okay, remember our bonding for these, right? Cytosine bonds to guanine, and adenine bonds to thymine. In RNA, adenine bonds to uracil, right? And you'll notice how I have different number of bonds here, right? Cytosine and guanine are bound together with 3 hydrogen bonds. So remember, that's hydrogen bonds that are between them, where adenine and thymine are bound together with 2 hydrogen bonds. So take a look at our picture over here, right? This is a nucleotide right here. Okay, I've got the phosphate group right there, right? There's my phosphate group. There's my 5 carbon sugar, okay? And there is my nitrogen base. And in this case, my nitrogen base here is adenine. Okay, this is a picture of DNA. So there's deoxyribose as the sugar. If it was RNA, my sugar here would be ribose. It'd have 1 more oxygen on it, deoxyribose, D, removal, right? So deoxyribose has 1 less oxygen. The phosphate would be the same, and if it was RNA, this adenine would be the same. And remember, DNA is double-stranded, right? So this adenine would have 2 hydrogen bonds over to a thymine that was attached to a sugar, right? That was attached to the deoxyribose and attached to a phosphate, because that whole thing makes up a nucleotide. So 3 parts to the nucleotide: phosphate, pentose sugar, nitrogenous base. Remember, thymine and DNA, uracil has RNA. We'll talk purine, pyrimidine. We're going to come back to DNA and RNA again, so we'll go over some of this stuff again. Okay, but these are the basic things that we should really remember from 9th grade. Remember, the order of the nitrogenous bases is what carries the information. So when it says the biological info carried in the sequence, remember that's the sequence of the nitrogen bases, the orders of our C's, G's, T's, and A's. Okay, when it says here they're linked by covalent bonds, remember it's the monomer that's linked by covalent bonds. So there are covalent bonds right here, here, and here, linking the different nucleotides together, okay? Not holding the 2 strands of DNA together. Remember, the 2 strands of DNA are held together by hydrogen bonds between the nitrogen bases. The one side of DNA, and remember RNA is single-stranded, those are held together by covalent bonds. Technically, those covalent bonds are called phosphodiester linkages. They're between the sugar and the phosphate. Okay, so again, I know most of y'all are currently in chemistry. So if you look at, let me just draw a different one over here so it doesn't get too messy. So if you look at your deoxyribose here, you'll notice how it has an O at the top there, but then it's just pointed. There's nothing there at the corners. So with this kind of drawing, each corner represents a carbon. So every time here, those are all carbons. Okay, and then you'll notice there's a carbon sticking up there. Okay, so that's where my 5 carbons come from, right? 1, 2, 3, 4, 4, 5. Okay, and so these will bond together. You notice how they bond the same way every time, right? The phosphate is on the 5th carbon, and the phosphate bonds to carbon 3 on the next nucleotide, bonding to carbon 3 on the next nucleotide. So one end ends up having carbon number 3 empty because it doesn't have another nucleotide after it, while the other end is called the 5' end because that 5th carbon that has the phosphate attached to it, that phosphate group isn't bonded to anything. Okay, and so we talked your freshman year about 3' and 5'. Well, that's what that means. The 3rd carbon is free or the 5th is free. And that gives that what's called directionality. Okay, and that directionality is important for how these are built. Remember, we're talking again, structure dictating function. The arrangement of these molecules is going to dictate how they behave, what they're able to do, okay, and how they work. And so this directionality, okay, you can only add nucleotides to the 3' end. So if this DNA strand that we have pictured here wanted to get longer, it could only get longer in this direction. Okay, and that's because of the structure of those monomers and how they're put together into the polymer. So that directionality becomes important when we're building more DNA because we can only add on the 3' end. And when we put 2 strands of DNA together to make a complete DNA molecule, those DNA strands become anti-parallel. Remember the strand running up this other side here would end with a 3' and a 5'. They run opposite one another. They run anti-parallel. Again, we're going to talk more about DNA and RNA again later. Okay, so let's look at proteins then. Remember, our monomer for proteins is an amino acid. Remember I told you amino acids are always going to look the same, right? They're going to have that NCC. They'll have that carboxyl group at one end. Okay, they'll have that R chain, which is what makes them different, coming off of that middle carbon every time. So when I form a polymer, protein is the polymer, when I link these amino acid monomers together, I do that with peptide bonds. Okay, and we saw how that was done with that dehydration synthesis reaction. You can see in this top picture here with the primary structure, you can see where this NCC pattern is happening, right? And every time you get that NCC break, that's a peptide bond. Those are different amino acids that are held together with those peptide bonds. There's my monomers put together into my polymer. Protein has, so when we link these amino acids together, that linkage determines what's called the protein's primary structure. Proteins have 4 levels of structure, and the primary structure is just the order of the amino acids. So this levels of structure for protein is new. We probably didn't talk about that in 9th grade biology. Okay, and so we need to make sure we get this down. Okay, so our primary is our order of the amino acids. That's determined by your DNA. It's determined by your genetics. Okay, and this primary structure, the way these amino acids are put together into the peptide with the peptide bonds into the polymer, okay, that also gives a protein directionality, just like the DNA had directionality. This gives the protein directionality. So you end up with an amino end or an amino terminus, and you end up with a carboxyl end or a carboxyl terminus. And just like in DNA and RNA, where I can only add more monomers to one end, I can only lengthen the polymer in one direction, proteins are the same way. That directionality then means that I can only add more amino acids on this carboxyl end. I cannot add any more on this amino end. And so that builds in, again, that directionality. The way these molecules are put together will start to dictate how they can behave. Okay, so I can only add more amino acids on that carboxyl end. Okay, so in addition to the primary structure of the protein, so again, the primary structure is the order of those amino acids, leucine, valine, glycine. Just like when we did protein synthesis in 9th grade and you just wrote down the amino acids in order, that's primary structure. So secondary structure, these proteins could be thousands of amino acids long. Okay? You can't have a big long molecule that's just that long in our bodies. We need it to get a little bit more compact. We need it to get a little bit smaller. And it's going to with this secondary and tertiary structure here. So the secondary structure is going to be due to hydrogen bonds along that phosphate backbone. Okay? So basically, you have these repeating structures, right? You have that NCC, NCC, NCC. So you have these repeating structures along the backbone of the protein. And so those repeating structures will form hydrogen bonds. Remember those hydrogen bonds again? Hydrogen bonds are between molecules, right? They are due to the polar parts of the molecule, right? The fact that one chunk of the amino acid will have a slightly positive charge while another part will have a slightly negative charge. And so those hydrogen bonds will cause the phosphate backbone of the amino acid. They'll cause the amino acid chain to start to fold in. And it can fold in one of two ways. You can get what's called an alpha helix or a beta pleated sheet. Okay? And within one protein, you might see both of these things happening. You might have an area of it that is alpha helixed, and then it switches to an area that's a beta pleated sheet. Okay? But those are going to be due to the interactions along the backbone. So you'll have the alpha helix and the beta pleated sheet. The tertiary level of structure, so the third level of structure, is going to be due to the interactions between those side chains. Those R groups, the ones that we said were different on each amino acid. So the interactions among those side groups is what's going to determine kind of the final structure of the protein. These could be hydrophobic interactions. They don't like water. They're moving a certain way to make sure they get away from it. Hydrophilic reactions, ionic bonding between organisms, or not between organisms, between the different molecules, between the different amino acids and the side chains. But it's the chemical properties of those side chains that are going to determine these interactions. For example, if you look at this polypeptide right here, how it's folding in on itself, these areas in here in the middle, those areas are, as a general rule, are going to be hydrophobic. They curl up into the middle because remember, everything in the organism is basically surrounded by water. They curl up in the middle to get away from the water. So the hydrophilic parts will be on the outside, the hydrophobic will be on the inside. But this final folding of the protein due to these different side chain interactions, hydrophobic, hydrophilic, ionic, disulfide bridges, these different interactions are what are really going to determine the final structure and function of the protein. The final structure will determine the function of that protein. Again, we'll talk about proteins some more in a little bit, but remember from 9th grade, we talked about a protein denaturing. If it lost its shape, it couldn't do its job anymore. That's because you break these bonds, it unfolds, and now it can't do its job anymore. The final structure is not in this final level, this quaternary level, is not in every single kind of, it's not in every single protein. These are interactions between multiple polypeptide chains. You see this here in this example over here is hemoglobin. Hemoglobin is what helps transfer oxygen in our red blood cells, the molecule that transfers oxygen in our red blood cells. You'll notice there are four proteins that all are coming together into one big one. That's the quaternary structure with the interactions between multiple polypeptides. So primary, secondary, and tertiary are in all proteins. The initial order of the amino acids, the folding along the backbone, and then the final folding due to the side chains. And then the quaternary structure is only in some polypeptides that interact with other polypeptides. Okay, two more and we're all set. Carbohydrates, complex carbohydrates, and lipids. So complex carbohydrates, so these are basically storage or building materials. Okay, and so for storage materials, we're going to look at two examples here, starch and glycogen. So right here in the middle, starch and glycogen. If you look closely at those, you'll notice that these are just glucose. Okay, so it's just a repeated chain of glucose. So here's my monomer, 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 monomer. So just a repeated chain of glucose over and over again. But if you look at how they're linked together, they're actually going to be linked together a little bit differently. They allow for, the starch and the glycogen allow for branching. When these get really long, they'll start to branch some, which allows for them to store more energy. These are storage molecules. Okay, and so the way they're linked together allows for them to branch, to store lots of things. Starch is storage in plants. Basically, they store their extra glucose like this in plants. Glycogen is storage in animals. Okay, mostly in humans, we store most of our extra sugar in the liver and in the skeletal muscles. And then when we fill those storage things up, we convert that sugar into fat. Okay, so glycogen is how we store it. Starch is how plants store it. If you look at cellulose, if you look closely, is also just a repeated glucose over and over and over again. Okay? But you'll notice the linkage of it is different. Okay? Basically, the molecule alternates between being right side up and upside down. It flips over every time it links together. And cellulose is a building material. Cellulose is found in the cell walls of plants. Okay, so it's found in the cell walls of plants. So if I'm making a building material that's supposed to make a wall, I don't want my wall to branch all kinds of wonky directions, right? I want my wall to be nice and straight and rigid. And so the way that the glucose monomers are linked together to form cellulose, the way that they alternate like that, is what causes them to make straight, rigid fibers. So where the storage molecules are branching to store extra material, these are going to end up in a nice straight line so that you get a nice, strong building material. Chitin as well. Remember chitin? Again, from freshman year, not pronounced chitin, but chitin. Right, so chitin is another complex carbohydrate that's a building material. Commonplace is the exoskeleton of animals, insects. It's found in the exoskeleton. Chitin is found in fungus. It's found in the cell wall of fungus. So it's a building material. So I want it to be, you'll notice the alternation, it's alternating, again, the linkage again. So I see the alternating linkage, so I get this nice straight line. So I get different properties. Because they were put together differently, I get different properties. And chitin is going to be a little bit different than cellulose because it's going to contain a nitrogen component to it. So that will make it behave just a little bit different too than the cellulose. So our last one here is lipids. Right? Lipids are non-polar macromolecules. So these aren't going to have a charge as a general rule. Okay, so there's not going to be any kind of charge here. How water had one part of it had a little bit of a positive charge and one part had a little bit of negative charge. Lipids will not have that. That's why oil and water don't mix, right? Because there's nothing for the water molecules to attract to, for them to bond to. So these are non-polar molecules. We're going to look at two examples here. We're going to look at fat. Fat's a very, very efficient form of energy storage. And this will become important later when we talk about we talk more about structure and function. So think about animals, right? Animals need to move around. They need to be mobile. They need to get from place to place. Okay? And so if we stored all of our energy in the form of sugar, we'd be huge. and it would be very difficult for us to move around. But since fat is more efficient form of energy storage, it's a more compact form of energy storage than carbohydrates, that is helpful in the structure of animals in enabling us to be able to be more mobile, easier mobile. Same with plants. It's more efficient and compact to store energy in fat. And so if you think about seeds and nuts are full of oil and fat, okay, because that gives a very good energy source to this growing, developing plant in a small, compact manner. So fats are a great, efficient form of energy. Okay, and fats can be classified as saturated or unsaturated. So if they're saturated, like let's look at this picture down here on the bottom. Okay, so the first one here I've got a saturated fat. The second one I have an unsaturated fat. So you'll notice the difference is this double bond here in the unsaturated fat. So saturated fats have no double bonds. Every place that a hydrogen can attach to the carbon, it's attached. So it's saturated because all the possible hydrogen atoms are bonded to carbons. So it's saturated with hydrogens. All of the hydrogens all the places a hydrogen could bond, they're bonded there. An unsaturated fat has at least one double bond. It could have more, right? It could have another one down here and we lose some hydrogens. Okay, so it could have more than one, but it has at least one double bond. So that means there's a spot where I don't have as many hydrogens as I could. Okay, and what that double bond does is it causes this bend. There's a reason why it's bent like that. The double bond causes the bend. You can see a double bond here causing a bend over in this phospholipid. And when you get that bend or that kink in this chain, this hydrocarbon chain, it makes it so they can't pack as closely together, right? Think about it. If I have these chains of fatty acids, I have these chains of fat, and they're nice and straight, then they could pack nice and tightly together. If they're not, then they get kind of separated out, and so they cannot pack nice and tightly together. So an unsaturated fat is going to be liquid at room temperature, like olive oil. Where a saturated fat, where it has all those straight chains there, those will be solid at room temperature. Think animal fat. Think if you've cooked some meat, some bacon or sausage or some kind of meat on the stove, okay, and that grease from the meat cools, okay, it'll cool into a solid. Okay, so those kinks and those double bonds, those are what keep those oils liquid at room temperature and those unsaturated fats. Again, this comes back to a different structure gives it a different property. Having a double bond makes it liquid at room temperature. The phospholipids, the other one we're going to talk about, these are what are called amphipathic molecules. They have a hydrophobic tail and a hydrophilic head. So let's think about what that means. Hydrophobic, right? If I'm phobic of something, I'm afraid of it. So a hydrophobic tail is going to be afraid of water because it's nonpolar. It doesn't bond with water easily. There's no charges there to attract it to the water molecules. So it's hydrophobic. The head is going to be hydrophilic because it's polar because it has a phosphate group. And that's a weird thing for a fat, for it to have this phosphate group. But that phosphate group gives it a little bit of a charge. Okay, and this is huge for the function of phospholipids because they have these polar and nonpolar parts. When they are put into water, they will naturally assemble into the bilayer, right? They will naturally form this bilayer that we get right here with our nonpolar tails in the middle and our polar heads on the outside. Okay, that's huge for cell formation. Okay, for the organization of the cell, the fact that it can create this, that it can have this barrier between the outside environment and its internal environment. Okay, but again, that comes from this slightly different structure, having this phosphate group on it, totally changing its function and the jobs that it can do. Okay, so we'll wrap this unit up with nucleic acids, and then we'll move on to unit 2 later.